Dear students, welcome to today's class where we will investigate the dynamic pathways through which stem cells transform into highly specialized cells that shape every tissue and organ in the body. We will uncover both the intrinsic genetic and epigenetic regulators and the extrinsic environmental signals that work together to control this process, from stem cell potency to precise lineage commitment. By the end of our session, you will be able to connect these molecular mechanisms not only to normal development but also to diseases, gaining a comprehensive view of how life builds and sometimes misbuilds itself. So today we are going to discuss the topic molecular regulation of cell differentiation. The major learning objectives of today's topic are to explain the concept and significance of cellular differentiation. We'll be distinguishing between different levels of stem cell potency, analyze intrinsic molecular mechanisms regulating differentiation, evaluate extrinsic molecular signals influencing differentiation, correlate molecular regulation with pathological outcomes, integrate mechanical and biochemical cues in stem cell fate determination. Cellular differentiation is the process by which an unspecialized stem cell develops into a specialized cell type with a distinct structure and function. It is central to multicellular development, transforming a single zygote into a complex system of tissues and organs. In adults, differentiation sustains tissue homeostasis by generating new cells for repair, turnover, or immune responses. Differentiation involves major changes in size, shape, membrane potential, metabolic profile, and responsiveness to signals. These arise from tightly controlled gene expression changes, regulated epigenetically without altering the DNA sequence. Metabolic composition also shifts. Stem cells rich in unsaturated metabolites gradually adopt lineage-specific metabolic states. In certain tissues, muscle, epidermis, gut, and nervous system, precursor cells undergo terminal differentiation. They permanently exit the cell cycle, dismantle division machinery, and activate genes essential for specialized functions. Potency of stem cells. Totipotent from all embryonic and extra-embryonic tissues. Zygote, early blastomeres. Pluripotent. Generate all adult cell types, embryonic stem cells or induced pluripotent cells. Multipotent. Produce multiple but related lineages. Example, hematopoietic stem cell. Oligopotent, limited to a few related cell types. Unipotent, generate one cell type but self-renew. For example, muscle satellite cells. In pathology, the degree of cellular differentiation is a key cancer grading criterion. Poorly differentiated cells often indicate higher malignancy and aggressiveness. Let's talk about overview of cellular differentiation. Cell differentiation is a fundamental process in the development of multicellular organisms, where diverse cell types arise from a single embryonic cell to form specialized tissues and organs. In humans, this process produces over 200 distinct cell types, each adapted for unique physiological functions. During differentiation, cells undergo profound changes in size, morphology, metabolic activity, and gene expression patterns. Despite carrying an identical genome, differentiated cells activate only those genes required for their specialized roles while repressing others. This selective gene expression results in the production of a distinct proteome defining a cell's structure, activity, and function. This differentiation is not a random process, but rather a highly regulated mechanism that ensures the orderly generation of specialized tissues, necessary for organismal survival and homeostasis. Moving on to molecular regulation of cell differentiation. Students, the regulation of cell differentiation involves a dynamic interplay of intrinsic and extrinsic factors. Intrinsic mechanisms are cell autonomous, relying on transcription factors, chromatin states, and regulatory RNAs, whereas extrinsic regulation depends on signaling cues from the surrounding microenvironment, growth factors, and extracellular matrix interactions. Intrinsic molecular mechanisms. Intrinsic regulation includes the genetic and epigenetic machinery within a cell that programs lineage-specific differentiation. Transcription factors. They are the master regulators that bind specific DNA sequences and either activate or repress the target genes. They serve as the primary switches in lineage determination, ensuring that only cell type appropriate gene programs are executed. We have myogenic differentiation factor, a master regulator of muscle cell differentiation. MyoD is a basic helix loop helix transcription factor and one of the most studied lineage determining regulators. Identified in 1987, MyoD is capable of converting non-muscle cells into muscle-like cells, underscoring its powerful reprogramming ability. It belongs to the myogenic regulatory factor family, which includes MYF5, myogenin, and MRF4. MyoD binds to EBOX DNA sequences within the promoters of muscle-specific genes, activating transcription and committing mesodermal progenitors to the muscle lineage. It plays a crucial role in myotube formation, muscle regeneration, and satellite cell activation. Mutations or dysregulation of MyoD pathways are implicated in muscle disorders such as muscular dystrophies and rhabdomyosarcoma, where normal differentiation is disrupted. The GTA transcription factor family. GATA, named for their ability to bind the GATA DNA sequence, itself is the name of the DNA binding motif. The GATA family of zinc finger transcription factors regulates lineage-specific gene expression by binding to the conserved GATA DNA motif. Each member of the family has unique developmental roles. GATA1, essential for erythroid and megakaryocyte development, regulating hemoglobin and platelet formation. GATA3 controls T-cell differentiation as well as roles in the nervous system and memory gland development. GATA4, critical for cardiac development and differentiation of endodermal tissues such as the pancreas and liver. Mutations in GATA genes leads to developmental abnormalities and cancers. Together, these factors orchestrate precise gene expression programs that stabilize lineage identity during development. PU.1 or SPI1, a master regulator of hematopoietic lineage commitment. PU.1, a member of the ETS E26 family, is indispensable for hematopoietic differentiation. It directs stem cells into both myeloid, that is monocytes, macrophages, neutrophils, and lymphoid, B cell lineages. PU.1 binds purine-rich ETS motifs to activate immune-specific genes. Importantly, PU.1 functions in a dose-dependent manner. High levels promote myeloid fates while low levels support B cell commitment. It operates in cross-regulatory networks, particularly with GATA1, where mutual repression ensures the lineage exclusivity between myeloid and erythroid cells. Beyond gene regulation, PU.1 also facilitates chromatin remodeling, creating open regulatory regions for transcription. Mutations or misregulation of PU.1 disrupt differentiation and contribute to acute myeloid leukemia, or AML, underlining its importance as a master regulator of hematopoietic cell fate. Moving on to epigenetic mechanisms regulating cell differentiation. Epigenetic mechanisms govern heritable changes in gene activity without altering DNA sequence. They serve as molecular memory systems, ensuring that once a cell commits to a lineage, it maintains its identity across divisions. Chromatin structure and chromatin remodeling. It enables dynamic regulation of gene accessibility. Two major processes mediate this: histone modifications by enzymes such as HATs, HDACs, and methyl transferases. ATP-dependent remodeling complexes which reposition or evict nucleosomes. Chromatin remodeling regulates transcription, replication, repair, and apoptosis. Dysregulation is strongly linked to cancers, making remodeling enzymes attractive therapeutic targets. DNA methylation. DNA methylation at SPG sites, catalyzed by DNMTs, repress transcription by reducing transcription factors excess. In pluripotent stem cells, pluripotency genes like OCT4 and NANOG are unmethylated, allowing expression. Upon differentiation, these promoters become methylated and irreversibly silenced. Failure of methylation disrupts lineage commitment and can trigger apoptosis. Because methylation is faithfully inherited during cell division, it ensures stable maintenance of lineage-specific gene programs. Histone modifications. Histones undergo multiple modifications on the N-terminal tails. Number one is acetylation. H3K9AC, H3K27AC promotes open chromatin and active transcription. Second is methylation. Context-dependent H3K4 Me3 marks active promoters, while H3K27 Me3 silences developmental genes. Number third is phosphorylation. Gamma H2AX, H3S stem involved in DNA repair and mitotic chromosome condensation. These marks act as a histone code, fine-tuning gene expression patterns during differentiation. Moving on to histone modification mechanisms. Acetylation neutralizes lysine charge, loosening chromatin and enabling transcription. Dysregulated acetylation contributes to cancers, with HAT, HDAC inhibitors used therapeutically. Methylation provides stable regulatory marks. For example, H3K27 Me3 temporarily silences lineage genes in embryonic stem cells, while H3K9ME3 enforces long-term repression. Phosphorylation marks DNA damage sites, gamma H2AX, or regulates chromosome condensation during mitosis. Moving on to non-coding RNAs. Regulators of cell differentiation. Non-coding RNAs add another regulatory layer. MicroRNAs. MicroRNAs are small RNAs that degrade or repress translation of target mRNAs. Examples include microR1, muscle differentiation, microR223, myeloid lineage commitment, microR181, T-cell maturation. Long non-coded RNAs act via chromatin remodeling, transcriptional control, or as competitive sponges for microRNAs. Examples are HOTAIR, which guides chromatin modifiers to silence developmental genes. LINC-MD1 promotes muscle differentiation by sequestering inhibitory microRNAs. Together, ncRNAs form intricate regulatory networks with transcription factors and epigenetic modifiers, ensuring precise temporal and spatial control of differentiation. Dysregulation contributes to cancers and developmental syndromes. Moving on to extrinsic molecular mechanisms. These are non-cell autonomous factors, signals from the environment or neighboring cells that influence differentiation. Extrinsic molecular mechanisms that regulate cell differentiation are growth factors and cytokines, BMPs, FGFs, TGF, beta, as regulators of cell differentiation. Bone morphogenetic proteins, BMPs. These are part of TGF-beta superfamily and are pivotal in embryonic development and tissue maintenance. They regulate mesodermal and ectodermal differentiation through SMAD signaling. Depending on context, BMPs promote osteogenesis, chondrogenesis, or neuronal differentiation. High BMP favors bone and cartilage formation, while inhibition promotes neural lineage. Second is fibroblast growth factors, FGFs. FGFs bind FGFR tyrosine kinase receptor, activating MAPK and PI3K AKT pathways to regulate proliferation, migration, and differentiation. They are essential in limb morphogenesis, angiogenesis, and neural development. FGF2 maintains embryonic stem cell pluripotency, while precise modulation guides neural and mesodermal fate. Number third, transforming growth factor beta, or TGF-beta. TGF-beta signals via SMAD3 and SMAD4 to control proliferation, apoptosis, and differentiation. In stem cells, it maintains pluripotency, but can induce EMT and mesodermal differentiation. Its effects are context-dependent, functioning as either a differentiation driver or suppressor. Cell-to-cell signaling pathways. Cellular differentiation is often governed by precise intercellular communication. Many of the molecules that mediate this process are growth factors, proteins or peptides, secreted by one cell to influence the fate of another. Although the molecular details of signal transduction vary among pathways, they share common principles. A signaling event begins when a ligand produced by one cell binds to a receptor on the surface of a target cell. This binding induces a conformational change in the receptor's extracellular region, which in turn alters the shape of its cytoplasmic domain, often activating its intrinsic enzymatic activity. The activated receptor catalyzes the phosphorylation of specific intracellular proteins, triggering a cascade of phosphorylation reactions. This chain of events ultimately activates a previously inactive transcription factor or cytoskeletal protein, leading to altered gene expression and structural changes required for differentiation. Importantly, not all cells are equally responsive to these signals. A property known as competence, which depends on receptor expression, intracellular signaling capacity, and developmental stage. Moving on to key signaling pathways regulating cellular differentiation. Number one is notch signaling. The notch is a signaling pathway, and it's highly conserved cell-cell communication mechanism found in most multicellular animals. In mammals, 4 notch receptors exist: notch 1, notch 2, notch 3, and notch 4, each being a single-pass transmembrane protein. The receptor consists of a large extracellular portion that associates via calcium-dependent non-covalent interactions, with a smaller segment containing a short extracellular region, a transmembrane helix, and an intracellular signaling domain. Students' notch signaling is activated when a membrane-bound ligand forms an adjacent cell, such as delta-like or jagged proteins, bind to the notch receptor, leading to proteolytic cleavage and release of the notch intracellular domain. Notch intracellular domain translocates to the nucleus, where it regulates transcription of target genes, controlling proliferation and differentiation. During neurogenesis, notch promotes proliferative signaling, maintaining neural progenitor pools. Inhibition by NUM allows cells to exit the cell cycle and undergo neural differentiation. Notch is crucial for embryonic development, vascular formation, and immune cell specification. Dysregulation is linked to cancers such as T-cell acute lymphoblastic leukemia, TALL, as well as genetic and vascular disorders like CADASIL, multiple sclerosis, tetralogy of F-ALOT, and L-Legile syndrome. Inhibition of notch has been shown to reduce proliferation of TALL cells in vitro and in vivo. Moving on to second one, hedgehog signaling pathway. The hedgehog pathway is another fundamental developmental regulator transmitting positional information during embryogenesis. The pathway is named after the hedgehog polypeptide ligand in Drosophila melanogaster, mutations of which cause larvae to appear spiny like hedgehogs. In vertebrates, the hedgehog family includes sonic hedgehog, Indian hedgehog, and desert hedgehog. These ligands bind to patched receptor, relieving its inhibition on the smoothened protein, which activates GLI transcription factors. This activation regulates genes controlling cell fate, pattern formation, and organogenesis. The hedgehog pathway plays essential roles in limb patterning, neural tube development, and somite differentiation. Malfunction contributes to diseases such as basal cell carcinoma, medulloblastoma, and developmental defects. Third signaling is WNT signaling pathway. The wingless, discovered in Drosophila, and INT1 were homologous genes. The name was combined as WNT. WNT pathway regulates both embryonic development and adult tissue homeostasis. WNT ligand binds to frizzled family receptors and co-receptors. LRP5, 6. Initiating a cascade that stabilizes beta-catenin in the cytoplasm. Stabilized beta-catenin translocates into the nucleus where it binds to TCF, LEF transcription factors to activate target genes. WNT signaling influences axis formation, stem cell maintenance and cardiac development. In adults, its dysregulation is associated with cancers, osteoporosis and degenerative disorders. Moving on to extracellular matrix cues as regulators of cell differentiation. The ECM is not merely a structural scaffold, but a dynamic signaling microenvironment composed of proteins, collagen, laminin, fibronectin, and polysaccharides, like glycosaminoglycans, proteoglycans. ECM molecules bind to integrin and non-integrin receptors, activating intracellular signaling pathways such as MAPK, PI3K, AKT, YAPTAZ, and Rho-GTPases that influence cell proliferation, survival, and lineage commitment. ECM mechanical properties, stiffness, elasticity, and topography direct stem cell differentiation through mechanotransduction. For example, stiff matrices promote osteogenic differentiation of mesenchymal stem cells. Soft matrices encourage neurogenic or adipogenic differentiation. ECM biochemical composition also plays a role in laminin-rich environments favor neuronal differentiation. Fibronectin or collagen-1 supports myogenic or chondrogenic outcomes. Additionally, ECM-bound growth factors such as TGF-beta, BMPs, and FGFs are presented to cells in a controlled manner, refining signaling responses. These cues interact with epigenetic mechanisms and cytoskeletal remodeling to program gene expression, patterns essential for differentiation. Dysregulation of ECM signaling can lead to pathological conditions such as fibrosis, cancer metastasis, or impaired wound healing. Integrin-mediated sensing of ECM stiffness as a regulator of stem cell differentiation. The extracellular matrix provides structural integrity and acts as an active regulator of cell behavior through both biochemical and mechanical cues. Among these, matrix stiffness is a key determinant of stem cell fate, sensed primarily through integrin receptors. These transmembrane proteins anchor the ECM to the actin cytoskeleton via focal adhesion complexes, enabling mechanotransduction, the conversion of mechanical signals into biochemical responses. In stem cells, integrin-mediated stiffness sensing strongly influences lineage specifications. Mesenchymal stem cells, for example, display stiffness-dependent differentiation. Soft matrices, 0.11 kPa, mimicking brain tissue, favor neurogenesis. Intermediate stiffness, 812 kPa. Similar to muscle, promotes myogenesis. Stiff matrices, 30 kPa, resembling bone-induced osteogenesis. These outcomes are orchestrated by integrin clustering and activation of downstream pathways, such as Rho-A, ROCK, FAK, and YAPTAZ, which regulate cytoskeletal tension, nuclear shape, chromatin accessibility, and lineage-specific gene transcription. So students, cell differentiation integrates intrinsic regulators, transcription factors, epigenetic mechanisms, non-coding RNAs, and extrinsic cues, growth factors, cell-cell pathways, ECM signals, ECM stiffness sensing via integrins is central to translating mechanical environments into stable gene expression programs, with critical implications for developmental biology, regenerative medicine, and tissue engineering. Dear students, together these discussions will equip you with a clear understanding of how molecular regulation governs cell fate, linking fundamental biology to its profound implications in health, disease, and therapeutic innovations. Thank you for giving me a patient hearing.